Okay, we're going to talk about mitosis, which is the exact duplication of cells. The reason I say that is that, as we talked about in the cell divisional cycle and cell divisional cycle control, that in mitosis, a cell divides, which is called the mother cell, into two daughter cells. Each are exactly the same. The reason I specified that, there's another process called meiosis, in which there's an inexact cell division. The two cells are not exactly the same. So mitosis is duplication of cells. We talked about the reason for it and all kind of different things. This involved the M phase of the cell divisional cycle. Mitosis is technically the duplication of the nucleus. That's what all these steps are. To duplicate the cytoplasm, which splits it, is called cytokinesis. Remember that everything is being controlled by genes and chemicals being produced to control cell division. So, a set of genes does mitosis and another does cytokinesis action. Therefore, you could do mitosis and not cytokinesis. If that occurs, and it does occur in some cases normally, then you would get a cell that's multinucleated because you would have divided the nucleus, but you didn't the cytoplasm. Okay? The stages of mitosis are prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Those are the stages of the M phase. You can look at these stages as we go along there. Prophase is the first stage. Prophase is a stage in which the cell does three things primarily. Dissolves its nuclear membrane, so thus it lets the genetic material out. The whole cell becomes the nucleus in a way, to give it room to divide adequately the nucleus. The genetic material calls from the loop domain, which you cannot see under the light microscope, to the chromatid chromosome fold, which you can, and it assembles a mitotic apparatus. A mitotic apparatus is basically a group of proteins that assist to move the genetic material properly. That's prophase. The mitotic apparatus is a structure that creates a framework forcing the separation of the genetic material. It helps elongate the cell also. The components of the mitotic apparatus are the centrioles, something called Kinetico Fibers, where the old name was chromosomal fibers, something called polar fibers, and also called aster fibers that some authors call aster rays. So these four components make up the mitotic apparatus, which acts as a group of spindles to assist the genetic material to move properly and to make the cell get longer so that you could divide the cytoplasm nicely if that occurs. Here, right here, these would be the microtubules. Here are the chromosomes. This right here would be the centrioles once they split. Those are aster fibers there that you see here. Here, right here, are these fibers. Those would be called chromosomal, but more appropriately called kinetico fibers, because they attach to the chromosome at a region where the centromere is, known as the kinetochore. The centrosome was discussed earlier, and what is a body in the center of the cell. So what the centrosome does, and the centrosome, go back to early one and read your text, is made up of two centrioles. What will happen is the centrosome has the ability, it has genetic ability to duplicate, and it will then take the centrioles and move them, once it duplicates, because each centrosome had two centrioles, it'll move towards the poles. Now, in mitosis, we look at it almost like the Earth. We have, this would be the equatorial region here, and these would be the poles here. So when you hear the poles, you mean in the corners, and this would be in the equatorial region, right there. Okay? So we go here. Here again was a centrosome made up of two centrioles, but it has genetic material where it can duplicate. So you would have two centrosomes, each having two centrioles. Okay. The centrosome containing the centriole acts as a focal point in the cell where microtubules grow out to help hold the cell shape. That was called the mitotic organizing center. In other words, this would be like the spoke in your bicycle, and these would radiate out to hold the cell shape. But they also can act in duplicating to help in mitosis. Here's the centrosome within a cell composed of two centrioles. These would be the microfilaments and microtubules that are being organized around the centrioles. Okay? Part of your cytoskeleton we talked about. The centrosome duplication occurs in mitosis. A cell generally has one centrosome containing two centrioles. The two are connected to each other by unidentified proteins. Duplication starts at the time of the G1S. Remember G1S. After the centriole does that, then they split and go to opposite areas. Right here, by them splitting, it rearranges the microtubules around them. Now, we do know even without it, it'll happen, but it happens better if you have the centrosome organizing, kind of like a drum major in a band to organize around. Here would be the centrosome composed of two centrioles, and now what has happened is duplicated and they're moving. But as they move, they start organizing these other microtubules. Now all of this is happening in prophase, the first stage. Right here would be the centrosomes with the two centrioles. And again, like I say, it's splitting. Now, we go to the Kinetico fibers. The Kinetico fibers hook to something called the centromere. The centromere is when DNA duplicates in a cell, it keeps the new strand attached to the old strand. It keeps the new strand attached for a while. When it does that, the two strands, the new one that was formed and the old one, are held by a centromere. Okay? The centromere is heterochromatin. It is not readable. Okay? Around the centromere are something called kinetico proteins, which are like a ring around the centromere. So technically what happens is these fibers called kinetico fibers attach to the centromere. To the kinetochore. But the kinetochore attaches to the centromere. We'll see that in a moment. Okay? So the function of the kinetico fibers are to push the genetic material in its proper location in the middle of the cell, the equatorial region, and later they're going to dissolve and pull on these two chromatids and break the two apart so that one chromatid will go this way and that way. Now the minute technically right now, that is one chromosome made of two chromatids. So my analogy of that would be, you are a mother, you have a baby inside you. You're getting ready to deliver. Right now, you are one person, theoretically. The minute 1 second later, when the kid is delivered, you're two people. Now, nothing changed in that 1 second except it was delivered. So right now, this is one chromosome made of two chromatids. But each of them is really a full chromosome. So what will happen once this breaks, the name changes because this now is a chromosome and that will be one. Just kind of read on it. Okay, here is the kinetochore kinetico fiber, microtubule hooking here. And it's being eaten away, which you'll see, to pull back and break the centromere. The polar fibers, on the other hand, overlap one another. They overlap. And they elbow out because right here there's an interaction between these two that elbows out and pushes the cell in an elongated fashion to make it longer so that if cytokinesis is going to occur, it would split it in the middle. Okay? Doing cell division, you create the poles. It also creates a section in the middle known as the equatorial region. The equatorial region. That's in the middle. The asteroids, their job is basically when you're trying to pull these things apart, you don't want to get pulled in. So let's say the asteroids kind of hold to the back of the cell and ensure you pull this way rather than this being pulled towards the center. Okay. All right. So you can read my PowerPoints. So prophase again, we said the nuclear membrane dissolves, genetic material coils, mitotic apparatus is formed. In interphase, you cannot see the genetic material using the light microscope. During early prophase, some divided into early and late prophase, the genetic material starts coiling and you can start to see it. See, here you really can't. It's grainy. All you see is a little heterochromatin. Now now it's starting to coil into this chromosome pattern, and you can start to see it under the light microscope. During the late prophase, the genetic material is coiled into worm-looking like microscope visible, which is the chromatid chromosome. Okay, we go further. Right here, mitotic apparatus forming. Right there, you can see this, the centrosome has been duplicated, and now the centrosome made of two centrioles are starting to move towards the poles. And the nuclear membrane is going to be dissolving. Here you can see the nuclear membrane is in fragments of the nuclear membrane. You've now started making the mitotic apparatus, asteroids, polar fibers, Kinetico fibers. Genetic material is coiled into a visible form in the light microscope. Metaphase's job is to push the genetic material into the equatorial region for adequate division. Metaphase, in that stage, is being pushed into the equatorial region for adequate division. It's pretty much the longest part of that 1 hour. The centrioles have moved to opposite poles. The centrioles have formed the mitotic apparatus, which is guiding the genetic material. And what you're trying to do is, when you open this out, you don't want the genetic material to get up to the corners. So these start shooting out and trying to grab the kinetochore to gently push these chromatid chromosomes into the center there. Okay. During metaphase, you get them into the equatorial region. At the high point of metaphase, listen to me, at the high point of metaphase, you will ultimately, using the Kinetico fibers, push it into the equator. See, initially I said the equatorial region. But during the high point of metaphase, the most prevalent point, you now have it right there on the equator. Okay? So when is the end of prophase going into metaphase? Prophase ends when you've gotten all of the 46 pieces of genetic material, all 46 pieces, into the equatorial region. When is the high point of metaphase? Is when it finally gets directly on the equator. When it finally gets directly on the equator. So we're going to end this one here and pick up mitosis video 2.